Okay, so we talked about the general structures of nucleotides, but now we have something a little bit different, but it's also similar but different. We need to talk about nucleosides and nucleotides. So there's the S here for nucleosides, T for nucleotides. So nucleosides, excuse me, nucleosides are just the sugar and the base. They do not have a phosphate on them, whereas nucleotides have the phosphate, have that third part. So nucleosides, let's look at those. So the base forms an N-glycosidic bond to C1. So we had a general structure in our last video up above on the page before, but now let's look at it more in depth and with real structures. So this position here, the 1' position of the sugar interacts with the NH on the N-containing base, and it forms a beta N-glycosidic bond. So beta meaning it goes up at the C1' position, and then it's connected to that base. So something that's important to remember is that this is going to be the reverse of a hydrolysis reaction. So we get rid of this OH, we get rid of this H here, we form water in this reaction and a nucleoside, because now we don't have the phosphate group on there, we just have the sugar and the base. So naming nucleosides is a little bit tricky in terms of remembering what things go with what. So the base names have INE at the end, except for uracil, which is CIL. Why it's different, I don't know. But to talk about the nucleoside you have that you have incorporated, you're going to switch the in these first two, you'll switch INE to OSINE. So instead of adenine, it would be adenosine. Guanine would be guanosine. And then cysteine, thymine, and uracil, it would be, you change the ending to IDINE. So cytosine, cytidine, sorry, and uridine. And then when you're talking about DNA, because you have the deoxyribose, you put deoxy at the beginning. So the same name, just deoxy at the beginning, deoxyadenosine. Continuing on, you'll see these X's here. That's because those don't occur in RNA when you have just your nucleosides. You don't have thymine in RNA, so we don't have a name for that. And then you don't have uracil in DNA, so we don't have a name for that. Okay, so then if we're going from a nucleoside to nucleotide, what we're going to do is have a phosphate binding to the OH at the C5' position of the sugar. So that is this position here. If you remember, we number, let's do a more fun color. If we number, we have 1', 2', 3', 4', 5'. Okay, so at the 5' position, we're going to make a phosphate ester. So we're adding our phosphate on. So in this case, it's adenosine if it's just without the phosphate. Then if we add the phosphate in, it's adenosine 5' monophosphate to show the full name. So this would be in the RNA version, right? Because we still have this oxygen here. So this would be the full name of the nucleotide associated in RNA. So in general, naming nucleotides versus nucleosides, all you really have to do is add the 5' monophosphate to the nucleoside name. So here's a chart here just showing you all of the different structures and names of each of the different monomers. So adenosine here, AMP is how you would put it in shorthand. adenosine monophosphate, that's AMP stands for. So this is the overall structure of that. If you replace this oxygen here with a hydrogen, you get deoxyadenosine 5' monophosphate, or lowercase d showing deoxy AMP. And then it's the same for the others. So we have guanosine here with the guanine base. We have cytidine here with the cytosine base, uridine here with the uracil base, and this one only has the deoxy form because it's T, so it's in DNA. So this table here is just for you guys to review, to look at the differences between DNA and RNA. So remember, our bases are slightly different, T and DNA, U and RNA, the other three are the same. The sugar in the structure of DNA is going to be deoxyribose and RNA is ribose. The nucleoside would be a base plus deoxyribose. Nucleotide would be base, deoxyribose, and phosphate for DNA. Same kind of thing, just ribose for RNA. And then the nucleic acid is going to be the linear chains of each of those groups. And then if you wanted, this is also a overall chart showing you the names for everything. So if you have a DNA nucleosides, DNA nucleotides, remember to make sure you add in that 5'. It's not in here, but you should have that. And then RNA having the base name, nucleoside, nucleotide. This is kind of a little cheat sheet for you guys here.